there is a pointer. Okay. Okay, thank you. Well, good morning, everybody. Wow, it is bright. I don't think anybody's mentioned that yet. So today I want to talk about some of the challenges we face in AAV phase 1 manufacturing and our approaches to meeting those demands. I'm going to go through this sort of as the story of us and our perspective on AAV gene therapy, because I think we have a unique one being in our phase 1 space, and some of the past, present, future challenges and our approaches to solving them. So I'm going to start off with just a set of basic trend lines that really, I think, are indicative of this is our evolution from our inception a decade ago up through the current date in 2018. And a couple of things to note in seeing these trend lines is the exponential growth in the past 2 years, campaigns, clients, manufactured vector, and personnel, and really the ballistic growth of manufactured vector that we've seen in the past 2 years, and in our efforts to keep up with client demand. And also that little bump in 2013 that you see in manufactured vector, what that represents, and really the significance of that bump in setting up the rest of the field to date. So a prehistory of us. We began in 1998 as a traditional research academic core facility with Dr. Reed Clark and Dr. Phil Johnson, who was the then head of the Center for Gene Therapy at Nationwide Children's. And that traditional core facility went on until 2004, and when Dr. Jerry Mendel took over the center, and he really had a vested interest in translational research and seeing some of the therapies that he was developing in the lab translated into phase 1 clinical trials. And at the time there was a real lack of manufacturing capability in the field. And so based on Reed's expertise in manufacturing and Jerry's desire to see these things into the clinic, they decided we could do this. We could make vectors, we could build a GMP facility and make vectors here, which is exactly what they did. And the rest is history, so they say. But as you can see by this, we really have our roots in an academic research environment through phase 1, and those roots exist today in our current mission statement of facilitating as many phase 1 clinical trials as we possibly can. So that legacy facility, 1,200 square feet, it was ISO class 7 BL2, designed around a producer cell line platform with cell carry space, manufacturing space, purification, and final fill space. So that the early beta platform we were talking about, that's stable producer cell line, scalability has always been a theme in our line of work, and we figured that was where we needed to start, but we figured out quickly to be able to handle multiple serotypes, multiple transgenes, this did not offer the flexibility. Very long lead times up front, 6 months or more in some cases, and in many cases it was virtually impossible to actually get a good producer cell line with the target VG per cell we were looking at. So we quickly moved to a transient transfection based platform, and where scalability was an issue here, we figured out how to scale it to a relevant scale that would facilitate the amount of vector we needed to make for these clinical trials. And we were averaging around 1.5 e to the 15 viral genomes in crude, but the trade in scalability was more than made up by the trade in by the flexibility. So it was where it took 6 months or more up front. Now it's 3 weeks. You can clone a plasmid in 3 weeks, and we could change serotypes, change transgenes just by swapping out a plasmid. To purify that vector, we were performing purification on cell lysates through microfluidization, taking it through clarification, TFF, iodixonol, chromatography, et cetera, through final fill, with 10 to 25% recoveries at the end of the day. And there was a major problem with that microfluidization step, is it didn't represent a closed disposable process, which represented significant regulatory hurdles. So we quickly moved to a media harvest, and where we now we're only harvesting roughly 70% of the vector, between 50 and 70%, as opposed to the 100% with the cell lysate. But the added benefit is we cleaned the feed stream up. So cleaning the feed stream translates farther downstream into actually about a 5% increase in recovery. So even though we're harvesting less, we were actually purifying more. So this is the platform that saw, you know, our first productions, you know, starting off with the toxicology prep and then going into AAV2 production in 2011 for an outside client in the UK. And 2012 seeing some additional campaigns for trials locally at Nationwide Children's Hospital. So here we are at that bump at 2013 and what that represents. And that's the SMA type 1 trial, the vector we delivered for that. And I can remember sitting down with Dr. Brian Caspar and Reed Clark and Will Fountain and having a conversation about what those vector demands were going to be for this trial, because now this was a systemic delivery, different than we'd seen before with, you know, IMs. And he was asking us to produce, it's somewhere around between 30 and 50 times more vector than we'd ever made before. And we knew immediately we couldn't just do 30 times what we're already doing. We had to figure out a way to increase our productivity, increase our scale, but not change, you know, keeping with the theme. We needed to be practical. We couldn't completely make a wholesale change. We wanted to make an incremental change that would make a big difference downstream. And that's when we discovered hyperstack technology. And this was a godsend for adherent cells, because you could in the same footprint as the 10 stack, it was 3 times the surface area. More importantly, you could manifold these things together and utilize or make one manipulation for 5, we call it a train, with 5 hyperstacks together. So where we increased our scale from 76,000 square centimeters to 360,000, previously we had 12 manipulations for that 76,000. Now we have 4. So we increased our efficiency immensely in this case, and without changing any of the base properties of our production platform. It's still a transient transfection, still the same amount of time, the same number of operators to do this. And then we also had to adapt our purification process. Everything else remaining equal, it's well known that iodixonol, ultracentrifugation gradients are a potential major bottleneck in scalability of AAV vector manufacturing, and this is, you know, so this is what we targeted to adapt to that scale, and we were able to do it by adapting a 2-stage iodixonol, and we were able to handle the increased volumes that were coming through with the increased feed stream. So another added benefit, we increased our recovery percentages by roughly 5% on the whole. And that's significant, and I'll get into that down the road. So we're into the current era, or the pre-current era, I should say, of 2015, 2016. We're still in our legacy space, but we realize, you know, with the success of the SMA type 1 trial, we got an influx of a lot of new clients that because there's still a lack of capacity in the field to produce, you know, phase 1 vector. And so a lot of extra clients and a lot of demand. You know, nobody ever asks for less virus. So it's something, you know, especially since they're going into showing that systemic deliveries work. So in our legacy space, we understood we needed to build, in order to meet this demand, we needed to build a bigger facility that could handle multiple products, and we started doing that 2015, 2016. But in the meantime, we were in this little, you know, 2-suite facility, And we had to do something to increase capacity yet again. So I'm going back to that Gen 1 platform just to show the timeline. We always looked at that 1 to 4 weeks as like every campaign traditionally starts with a thaw, scales up, and then goes into the actual manufacturing steps. So we thought, well, okay, why do we have to do that? We have an isolated cell carry space. Why can't we just, and the bottom half of that slide shows the expansion into 10 stacks, why can't we just carry cells continuously at that 10 stack stage and cut off the top half of that timeline and basically be at scale all the time, which is exactly what we did. And that enabled us to basically double our capacity in that single use, we had to line everything up in a row facility. And that carried us through until 2017 when we took over our new facility. So 2017, 18, 19, current era, current manufacturing, now we're into that exponential growth. As soon as we built it, they came. We were immediately at capacity, and you can see here where, you don't have to read all of this, but we were doing a lot of work, and we were still struggling to keep up with our clients' demands and needs, for timely vector delivery of large quantities of AAV vector. So in 2016, we began construction of the new facility to support that multi-product manufacturing, as well as capacity for cell-based therapy, which is another space we realized was growing and a need for, and that we wanted to grow into as well and incorporate that into our GMP space. So we occupied that space in 2017, which was 9,000 square foot, 7,800 dedicated to vector manufacturing, including QC analytical space. 1,200 square feet of it was for the cell-based therapies initiative that we wanted to start, and the final design accommodated, like you said, 4 viral production operations simultaneously. And we also designed this facility to be flexible and adaptable to new technologies. We didn't want to bolt anything to the floor. We didn't want to be married to any one process. We learned that early on, designing our facility for one process and then quickly having to move to another. Here's a diagram of our new facility. So it consists of that central cell carry space, which still to this day, that's the way we're able to turn over so many different campaigns, is the fact that we continuously carry at scale. And that one small cell carry space is able to feed all 3 of the manufacturing rooms. And you can't read this either, but we so creatively named them Processing Room 1, 2, 3, and 4. So processing rooms 1, 2, and 4 are all manufacturing spaces, and then processing room 3 is dedicated to purification and fill. And you might ask, why only one room for purification and fill? The reason being is the manufacturing step tends to take much longer in most cases than the purification and fill steps. So we're able to leverage that one room for all 3, but you'll also notice that processing room 1 is bigger than the other 2 manufacturing spaces, and that's because in that room we're able to carry a process all the way through from manufacturing through fill. So we didn't want to lose redundancy, which is another thing in manufacturing that's critical and key, is always be redundant. Always have redundant processes. You never know when an ultracentrifuge is going to go kaplooey, and we've seen it all happen. So this space also includes research in QC lab spaces. The QC lab serves the GMP manufacturing space as well as the research and academic manufacturing space, and also our PD, which is, we also wanted to build a process where the testing was consistent across every facet of manufacturing, and that's why we did this. And then there's that cell-based therapy space that our former director designed around his experience in industry. So like I said, we built it and they came, and we still struggle to meet client demands as this field continues to explode. So what's the easiest, most practical thing to do is just, in this case, we were able to just do more of what we were already doing. We expanded our capacity by 50%, adding 2 more trains to the process. All you had to do was add 1 more incubator. Didn't change the process. It was still the same timeline, same platform, and same number of operators. It just added 50% more capacity. Basic snapshot of our process. That continuous, as it is today, is our continuous seeding process in cell carry space. Weeks 1 and 2 start at transfection, or actually terminal seed through transfection and harvest. And then weeks 3 and 4 are gradient ultracentrifugation. Starting with gradient ultracentrifugation occurs in that processing suite. Weeks 1 and 2, we can do multiple times over. So that's how we're able to facilitate large vector demand campaigns, is by doing multiple sublots, basically repeating in weeks 1 and 2. So, now 2016-17 saw us growing and actually becoming a full-fledged CMO, even though, but we keeping our roots in that academic research core facility, and and with the serving our mission to facilitate as many phase 1 clinical trials as we possibly can from our position. We found ourselves growing into a full-fledged CMO that with a full complement of employees. So now we're over 80 employees, where we started with 6. But the notable thing about that is if you, and again, it's probably too small to read, but our QC team, led by Will Fountain, was one of those original 6. Lenore Giannunzio now heads up our process development team and cell-based therapy, our non-viral initiatives. She was part of that original 6. Myself, and I was part of the original 6, and then Barigila DeWalker, who heads up our quality team, she was part of that original 6. So, aside from Reed and 1 or 2 others, the core of that first facility still exists and represents the leadership of our current team, and I think that's not insignificant. So, from those first days of trials that, literally the building beside us, to the one trial in the UK, we now have a global impact. We see, from Canada to New Zealand and everywhere in between. And go back. And as Henry was saying, we've supported over 40 successful INDs, EU, rest of world trials, either directly as a CMO, or directly in our organization as an institute or as a CMO. So that represents a lot of current challenges that we're working through. And one of the first, the most obvious is just managing that growth. And the problem you have is as you grow, you can't grow any part of the triangle without growing the rest of the triangle. As your GMP production capabilities increase, your quality systems and your analytics need to grow along with it to keep up with that demand. Your PD is also has to grow, because as Luke was talking about, all the different engineered serotypes that are being developed now with improved tropisms, et cetera, come along. We have to come up with clever ways to purify them, and then research production sort of by proxy grows right along with everything else. And ironically enough, it's the research production that is growing the quickest among all of these groups. And if you think of that triangle as sort of the tip of the iceberg, what lies beneath is supply chain and logistics. And in our critical reagents like hyperstacks and plasmids and things like that, as we grow, the field is growing along with us, and those we're stretching suppliers to the limit at this point, and that's where the critical and very good relationships, like the one we have with Aldevron, is it becomes ultra critical to manage this growth going forward. And then the third bullet point here is one of the things I have to admit we really didn't see coming, and that's management of worldwide transfer of product. We never saw ourselves as a distribution company, and we still try not to be. But with a global reach in terms of the trials we're trying to facilitate through our manufacturing, we find ourselves transferring product, finished product to different, all over the planet, as well as managing shipments for QC testing and stability testing, et cetera. And another thing is client volatility. We tried to make a process that was consistent from design and proof of concept. This is sort of trying to depict a pathway here. Design and proof of concept through research grade production, preclinical studies, tox vector, all the way through GMP production of your phase 1 and beyond. And the reason being is because consistency is key in these cases. It's, your tox vector must be made under the same conditions and manufacturing platform as your GMP. So it pays dividends if everything can be made the same way from the early on of the life cycle to the late, all the way through the end. But every client is different, and every client comes to us in a different point in this pathway, and there's major scheduling implications that occur as a result, and every one of them has different yield demands, et cetera, and their own goals and timelines, and some have patients already lined up, et cetera. So we have everything from a grandmother, and this has happened. A grandmother called us who has a child with an ultra rare congenital disorder that has found a PI that has a potential therapeutic, and they really don't know where to go next. And so they've called us and said, what do we do? And we've helped them all the way through a top 5 pharma company that has a very mature product that they're just looking, but they're not in the business of making phase 1. So they're just looking to us to be a CMO for them for that phase 1 step. So as you can see, it lends to a great deal of volatility in our queue and a significant challenge. Capacity is another issue. I mean, I can't mention this enough. Even though we've increased our capacity, we plan to increase our capacity again in terms of the adherent system and add more hyperstacks to the sublots. But we also see a need to go to a new platform in a suspension cell line and a bioreactor. But we didn't want to do this until it was practical. Detect a theme. And so we set a bar. We said, we're not going to implement this into any of our facilities until we're at least at the equivalent productivity as a hyperstack. And luckily, Lenore Giannungio's team and PD worked very hard, and they've reached that equivalency step. So we are now implementing a bioreactor process as part of our platform. So the future, 2019, 2021, and beyond. What do we see coming down the road in the field? And one of the future challenges we're seeing, especially with this changing regulatory landscape, where it's getting quicker and quicker to go into commercialization with any given product, is having a commercial-ready platform. The hyperstack platform we think is very appropriate for certain scales, certain indications like ocular indications and low dose indications. It's very practical. I mean, I'm way oversimplifying this, but you put cells in it, in media, the cells grow, you transfect it, it makes virus. Again, way oversimplified, but it's not too far off. It's not nearly as tricky as a bioreactor, so it works very well. But in many cases, we have to go into to make something commercially viable. Our clients are going to have to make that jump to a bioreactor process, so we endeavored to make where we knew we were creating a gap there, because for those that are able to start in the process and move on in the same process, they're able to maintain that consistency that's so key. But some we knew would have to make a jump at some point. So we tried to make the bioreactor process as close to the adherent process as possible. So it's still HEK293 cells. It's still a transient transfection. The downstream processing is essentially the same, and we've actually figured out ways to increase our throughput through gradient ultracentrifugation again. So where we knew we created a gap, we also tried to create as short a bridge as possible across that gap. And then again, larger yield demands, that ballistic curve of a manufactured vector and nobody wanting less virus. So we continuously try to increase recoveries. That's I think Luke was also mentioning yesterday that we need to increase our manufacturing capacity by tenfold to meet a lot of these demands, and I completely agree with him. But one of these ways is not just by increasing the amount we can manufacture, but the amount we purify. If you think about the fact that we lose 75% of everything we make, that's tragic. And there's new technologies coming out that are very hopeful in increasing those recoveries and still getting the type of empty to full ratio enrichments that we look for. And we continue to vet those, and we will implement them when it's practical to do so. Finally, plasmid design. This seems, a lot of people think of this as an afterthought, and because a lot of PIs at the very beginning of this process aren't thinking of, well, I'm going to take this to a commercial process. So we want to where we have a vested interest in the middle of the process. We also have a vested interest in the very beginning in that vector design phase. Not only to make sure that that our clients are getting the expression levels that they need out of these plasmids, but also looking with an eye for productivity. Are the ITRs integral and complete? Is it in an oversized backbone with the proper antibiotic resistance? All of these things we've seen, because most of the times it's a PI that's just trying to get some data and just trying to get some vector manufactured in that research, when they're in proof of concept phase, and they don't think about these things. And you think about it, once you start down that pathway, you're kind of locked in. So we want to get involved earlier on in the process. So in closing, the things that I kind of want to stress is, in the field, we want to make incremental changes that are significant and not necessarily reinvent the wheel, because we need to maintain that consistency through our manufacturing steps. And we want to leverage new technology whenever it's appropriate and practical to do so. We don't want to reinvent the wheel every time. We look at those shiny new things out there and vet them, but we don't implement them until it's time to do so. And the other thing is identifying efficiencies wherever they may hide. A lot of times that, just from an operational perspective, that is what really enables us to drive those yields up in a significant way, is just by finding the little things, the little efficiencies that allow us to do just a little bit, work a little bit better, a little bit faster with a better quality outcome. So that's the end of my story. Happy to take questions.
Thank you, Andrew. And yeah, it's not really possible to see if people have questions, so feel free to speak up. So let me get this straight. Exponential growth of people, need a new facility, need closed systems. I mean, that story sounds awfully familiar, yes, right? So,
a lot of parallels, right?
A lot of parallels there. So I also like you coined a phrase, client volatility, and I'm glad you coined that phrase and not me, but that's sometimes real, that's true, right? So that's really related to my question, not to put you on the spot in terms of your queue time, but maybe some comments for the audience and the new norm of what people should expect, or how should people plan for their overall supply chain to get their products into the clinic? How far in advance do you recommend they come and talk to you? How quickly do you sort of want them on your schedule to make sure, that they're not stuck and in this new norm? So any comments about turnaround times, queue times?
So we're actually queued, to talk about our queue, we're booked through 2019. So that's why we're increasing our capacity yet again, right, to try and compress that to allow for more queue space. But yeah, if you have an idea, come talk to us yesterday. You know, that's the earlier on you can get into that pathway, the better off you are. You know, we queue based on, you know, we have to do it sort of as a first come first serve basis, and first one ready to go. These are all phase 1 trials, and they're all fraught with danger. You know, in terms of, you know, we've seen lots of things happen. So, you know, we tend to queue people up and then say, this is our first available space, and we do everything we can to have them ready to go when we get there. But sometimes that's not the case. So we'll end up sliding them back, but we're able to, at that point, in most cases, slide somebody up.
Right.
So, you know, there's this constant, we call it playing Tetris, this constant game of Tetris that we're doing with our clients, and there's a great deal of volatility, and it could change on a daily basis.
Know the feeling. James. Oh, sorry.
Hi, I come from a similar, not the same type of environment, but a hospital-based environment where we have a large virus group making oncolytic viruses, and I run the cell therapy part. So I was just wondering, from an administrative point of view, how do you see working together? Is it in that academic structure, is it all administered as one? And are you competing with each other for space and resources, or have you come to some sort of...
No, I think we've come, I mean, our administrative is extremely supportive of us, and this cell-based therapy initiative is new. So we're kind of working through that, but we're trying not to, we're not really competing for space. There's, you know, defined space in our facility for cell-based therapy, you know, and we're trying to meet the staffing demands and everything else as that comes along. We have a separate leadership group that's sort of driving those technologies into our cell-based therapy space. So it's, we haven't seen what you're talking about just yet.
Well, we found it initially as well, because we were a little bit behind in terms of the GMP aspect, and the virus people were able to help us get some of those SOPs and everything in place.
Oh, absolutely.
Right? So there's, but now it's down, we're sort of competing for resources at this point. You know?
Yeah.
So
yeah,
and I don't know what the right administrative structure is to oversee that. I think we're coming together a little bit as we move more to exosomes and various other things. Many of the processes are going to be starting to look somewhat similar between the two. So again, we could be back where we can sharing and learning from each other again.
Yeah, I agree. You know, we did the same thing. You know, we leveraged a lot of our previous knowledge from vector production into the SOPs and the whole GMP structure. It's under the same quality structure as our vector manufacturing and everything else. So, you know, we're trying to maintain a synergy there, and as well as with staff and with technology also. So, Dr. Dennis Durbin, who our Chief Scientific Officer, is here, and if you want to talk about administration, I'm sure he would answer your questions also.
Yeah, just a real quick one. Thanks, that was a great talk, Andrew.
Thanks.
When you look at that facility and the floor plan and whatnot, my question is, do you run more than one campaign at a time in there? And, you know, what are the sort of concerns about doing that, just in general?
Sure, yeah, we do run more than one campaign. So we run, you see there's three manufacturing suites. We're running 2 of those suites simultaneously, with the other suite staggered, on and off staggered by 1 week. And we're able to do that because we have a very efficient and thorough changeover process from one campaign to the next. Also, like I was mentioning before, we're typically in multiple sublot campaigns. So one room will be running a single, one room will be running one product for an extended period of time, and the next room will be running a different product for an extended period of time. Each one of our rooms are isolated by differential pressure, differential air pressure. So, you know, we're able to isolate products by engineering through air pressure, as well as facility behaviors and flows. By the way, our personnel are able to move in and out of rooms and not into other rooms.
Yeah, along those lines, what's the state of sterile filtration in the AAV regulatory and process development landscape?
Well, it's a critical step. You know, that's so. I mean, you must do it. So maybe I'm not understanding your question completely, but we have multiple filtration steps throughout the process. I mean, we don't see bioburden a lot, but you know, just in case it's there. There's multiple steps along the process, and there's a step just prior to final fill. This is one of the major EU regulations that we follow, is you have to sterile filter immediately prior to fill. So, I mean, that is what we consider a critical step in the process.
Hi, Andrew. You
Hi.
talked about plasmid design at the end of your talk. How resistant are your customers to reconfiguring plasmids, and is there a point of no return within the process?
There is sort of well, there's never a point of no return, but there's a point of great pain. Once you get beyond that, basically that pivotal tox study, that is your pivotal study to go into phase 1 manufacturing. And once you get there, you're sort of locked in without having to do significant bridging. So you can still do it, you can still change, but it requires a very robust bridging study in order to get over the regulatory hurdle. You know, I would say as early on as you can lock yourself in, the better off you are. And you talk about resistance to change. Everybody's different. Every client has different, you know, and honestly, you know, if they don't approach us about it, then, you know, we're fine with that. We'll move forward with it, whatever you want us to move forward with, and, you know, and we'll go from there. But in some cases, you know, you have clients that are insistent to move forward with a given plasmid. It doesn't, you know, it'll prove itself out. You know, in research productions, et cetera, it'll just show that it doesn't yield, it doesn't produce, or there's a great deal of variability. We've seen this. And then, you know, sometimes it takes a little convincing. They go back, do the recloning, and then we've seen up to a 4-fold increase in yield productivity, just based on, you know, being in the right backbone and with good integral ITRs, et cetera.
One more question.
Hey, Andy, it's Ken Leclerc. I don't know if you can see me or not, but I may not have worked at a company that we worked with you, but I can attest that these guys bend over backwards to help pretty naive clients, so great job. Question about the producer, suspension culture, serum-free. As you go to that platform, you're going to put more pressure on plasmid supply. So it's a good thing Aldevron's had the foresight to build a big building a few miles from here, so we really knew the producer cells to come along. Any comment on nanoplasmids? Because there is vector design is a big part of it,
Yeah.
and reverse packaging of the backbone is a concern. So nanoplasmids are showing some good results. Have you played with those yet?
Haven't played with them yet, but this is something we've seen. I had a couple of articles and forwarded to me just last week about these, and we haven't put them to the test yet. But it's like you said, we're always trying to leverage new technology. We're always looking at what's next, and, you know, we'll absolutely evaluate them and see what we think. Mm-hmm.
All right, one more round of applause, please. Thank you.
Thanks.
Thank you very much.
Thank you.